This is the lecture for blood physiology. The purpose of this lecture is kind of threefold: to think about the physiology of individual types of cells that can be found in our blood. That also helps prepare us for immunology by also revisiting Bio5 and anatomy. So some parts of this lecture might be a review to you. The mechanism of clotting or hemostasis is going to be key for one of the written questions for the next exam. So that is probably new material for a lot of you. And thinking ahead to respiratory physiology as well as homeostasis in terms of how blood acts to carry different components is also going to be touched upon at the end of this lecture. We've already kind of talked quite a bit about the structure of these different learning units. Remember that we talked about the overall system of the cardiovascular system in terms of the anatomy review of blood vessels on the heart. We delved into the physiology, cardiovascular physiology, thinking about electrocardiograms, the cardiac cycle, hemodynamics, and clinical applications. And now we're kind of zooming in and thinking about the actual material that gets carried around by the cardiovascular system. So while the cardiovascular system's purpose is to move blood where it needs to go, the blood itself has its own function. So we'll be thinking about the physiology of blood and lymph. Again, today we're thinking about the function of individual blood cells, how clotting occurs to prevent leaking out of blood, as well as oxygen saturation. In terms of the learning objectives for this part of the lecture, you should know about erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. These are different formed elements, so they're an important part of the connective tissue that is blood. We'll be thinking about how they're formed, the rough replacement rates, how long do they usually live for, the overall kind of structure and function of these erythrocytes, leukocytes, and platelets. We also want to think about the mechanism by which those leukocytes are leaving the bloodstream and getting where they need to go. That ties into the lymph and immunology lecture in terms of figuring out how cells are called to different locations of tissue damage. So I want to emphasize there that we're thinking about tissue damage broadly and preventing infection. The reason for that is because when we think about the process of hemostasis or clotting, that is answering what happens when we have vascular damage. So specifically, when we damage our blood vessels, how do we go about clotting and fixing that damage? So there's going to be a couple different mechanisms here. One is broadly tissue damage and introducing pathogens. The other is vascular damage. We'll also kind of think about clinical applications such as a white blood cell count and how blood typing is carried out. That how blood typing is carried out is something that you'll also delve into a lot in lab, this is lecture, during the blood lab. So in thinking about the physiology of individual types of blood cells, remember again that we started out by talking broadly about the cardiovascular system. So we have that cardio part, the heart, which is pumping blood, and the vascular portion, the vessels, which are moving blood. Remember that the surface area to volume ratio of the vessels changes as we move away from the heart and then return back to the heart. The reason for that is to maximize surface area and room for exchange between blood and the surrounding tissue. And the purpose of that is because blood carries so much stuff, it has a lot of important functions and it has to be able to interact with all kinds of body tissue. So when we're thinking about the structure of blood itself, remember that blood, we don't maybe think about liquids as being tissues, but they definitely are. And blood and lymph are examples of fluid connective tissues. So the thing that kind of ties all connective tissue together, it's very messy, there's lots of different types, but all of them have some type of unique cell type that is dispersed in what we call an extracellular matrix. That's some grounding substance that brings all those unique cell types together. It connects them. It's connective tissue. So it can be fluid, it can be solid, it can have different types of fibers in it. So things ranging from bone to cartilage to blood and lymph to different types of fat. All of those are referring to these connective tissues. So fascia, tendons, skeleton, blood, and lymph are all common examples. So we'll be thinking about kind of the role that the skeleton plays, the bone tissue plays in this as well. And remember that each of those different cells in bloods have distinct functions. So we'll think about kind of blood broadly, how it is produced, and the functions of those individual cells. So remember, again, blood is a connective tissue. There is a fraction of it that is plasma. Another fraction of it is formed element. So the plasma is that liquid, fluid, grounding substance. Those formed elements are similar to cells. Some of them are cells. There's the leukocytes and the erythrocytes. Platelets are not technically cells. They're fragments of cells, but they do come from a cellular source. So if we're just focusing on that grounding matrix, the plasma, and kind of thinking about the fraction of the overall blood that is the plasma versus the formed elements are measured according to what we call the packed cell volume, and we more specifically measure the hematocrit, the proportion of the overall blood, the total volume of blood that is actually red blood cell. So depending on your biological sex and a lot of different factors, a normal hematocrit range can be 37% to 52% of the overall volume of blood belonging to those packed cells or those red blood cells. So when we're thinking about this idea of viscosity, that's something that we mentioned when we went into hemodynamics and thinking about blood flow. It's kind of like how syrupy, how sticky and thick the blood actually is. So the clinical test for this is a hematocrit. That is defined as the percent volume of red blood cells out of the total volume of blood. So for example, if your hematocrit is 37%, that means out of the total volume of blood, 37% of that is red blood cells. So when we're thinking about those different layers, if we take a blood sample, run it through a centrifuge and kind of size fractionate or density fractionate it out, at the very top, the least dense is going to be plasma that has water, proteins, nutrients, and hormones in it. There's a kind of fluffy, buffy, bubbly coat called the buffy coat that has white blood cells and platelets, so formed elements that are not red blood cells. And then we have the hematocrit. That is the fraction that is actually erythrocytes, red blood cells. So we can kind of look at what we would expect a normal range to be and see what is maybe too low or too high of a hematocrit. Again, this kind of changes based on your biological sex, but when we're looking at the normal hematocrit range, that can be anywhere from 37% to 52%. So if we have too much plasma and not enough red blood cells relatively, we have a depressed hematocrit, a lower than baseline hematocrit, and that is considered anemia. There's a lot of different mechanisms by which anemia occurs. So there's iron deficiency anemia, there's sickle cell anemia. So there's lots of reasons why we might have a depressed hematocrit. So that tells you that anemia is kind of like a classification of clinical results or of overall pathology. It's not really telling you the mechanism or exactly what's wrong with the patient. Polycythemia is the opposite problem. It's when we have a very elevated hematocrit percentage. That's when blood viscosity is too high. And really what we're thinking about here is a situation in which we don't have enough plasma. So you might not think that having a high hematocrit is a bad thing, but it can be if it changes the viscosity of our blood too much or if it represents very severe dehydration conditions. So if our blood is too syrupy, that decreases flow and we're not getting blood where it needs to go. So when we're thinking about the overall function of blood, not just the individual cells, but why we have blood more broadly, the classical example is transportation of respiratory gases, oxygen to body tissue, carbon dioxide away from body tissue, so removal of waste, and then also bringing nutrients that have been absorbed or biosynthesized in some way where they need to go. So that's transportation, getting stuff where it needs to go. There's also defense. So we think about white blood cells, platelets for clotting, different proteins, the movement of cytokines, the signaling molecules. All of those are crucial for making sure that we are dealing with portals of entry and pathogens the way that we need to. And that's what's being shown on this GIF on the right. That is a white blood cell using CHEMOTAXIS to follow the chemical trail of a bacterium. It's able to then adhere to, engulf, and break down that bacterium. So that's an important role of blood, is the movement of defensive components. Also maintaining homeostasis. So we know that homeostasis covers a lot of different things. One is temperature, thermoregulation, the movement of blood, maybe keeping it deep within our body or taking it more to the surface so that heat can be given off. That helps regulate our temperature. It also helps regulate our pH. So we're going to talk later in the semester about acid-base balance, kind of thinking about the role that the lungs are playing in this and the role that the kidneys are playing in this. In the hemodynamics lecture, we talked about how an active cell is producing a lot of carbon dioxide and lowering pH. A more inactive cell is not producing as much carbon dioxide and has a more neutral or raised pH. So we're thinking about blood acidosis and alkalosis, changes in those pHs and how they're kind of adjusted. That is more considering respiratory acid-base balance. That we'll also talk about renal or kidney-related acid-base balance, which is called metabolic acid-base balance. And then blood is also crucial for regulating water content, so osmoregulation. Again, those anemia and polycythemia values often represent changes in the plasma fraction, that water content. That is also a huge thing that we're going to talk about when we cover homeostasis and excretion in terms of blood filtration. So going back to that image that I showed you earlier, we will be expanding on those formed elements quite a bit, but the plasma itself has a lot of important stuff in it. So just focusing on the plasma and thinking about specifically the plasma proteins, you might have heard of bovine serum albumin or egg albumin. Albumin is used a lot in microbiology and in molecular lab settings. It's also used in a lot of medication as kind of a stabilizing or buffering component. So what it is a binding protein. Remember that if something is fat-based, it doesn't really play well with the cytosol or the extracellular fluid. So this is basically a chaperone. It goes and it picks up the fatty acids or the steroid hormones and gets them where they need to go. It also helps maintain water in blood. So it's kind of like an Uber driver. It picks up those things that can't get somewhere on their own and takes them there. There's also globulins, which we cover in depth in the immunology lecture. Globulin is a very diverse group of protein, and remember these words end in IN because proteins usually end in IN, and so these are again plasma proteins. Globulins is a really big umbrella category. It includes transport proteins again, maybe for important vitamins, and it also includes immunoglobulins. So that is a fancy term for antibodies, or a more technical term. So when you see terms like IgG, IgM, IgD, those are all referring to these immunoglobulins, the antibodies. And then one more that I wanted to touch base on is fibrinogen. That is a protein that is involved in blood clotting, and it gets activated to fibrin. So while those proteins are found in the plasma portion, that ground matrix, the fluid portion of blood, the formed elements are formed through a process called hematopoiesis. So that's a general term to refer to the process by which our body is making blood, and this physiological process changes over time. So specifically in terms of the location in which this is happening, it happens in dramatically different places during fetal development and then in our early life. So generally we think about hematopoiesis as happening in the bone marrow. We think about development as happening in the bone marrow and also in the thyroid. or sorry, in the thymus. So we have these different locations kind of in our mind of where that development takes place. Early on in fetal development, we don't necessarily have those structures. So initially, hematopoiesis happens in the yolk sac and then in the liver and the spleen. Rates of that happening in the liver and spleen kind of drop off prior to birth, but we call that extramedullary. You should know from anatomy that the medullary cavity is a structure in bone. Extramedullary means outside of the bone. So that happens in the liver and the spleen, but most of it is happening by the time we're born in our bone marrow, because at that point we do have bone marrow. Remember that bone changes quite a lot from the time we are born until we start to have more calcified bone in adolescence, and it continues to be remodeled throughout our lives. So your entire skeleton replaces itself about every 10 years or so. But early on in development, there's a lot of that red marrow, and so there's a lot of hematopoiesis happening. And then as we kind of get older, there's more of it happening in different locations. The bulk of it is happening in the vertebrae, also the sternum and the ribs, and then it drops off in the tibia and femur as we get past about age 25, and then it still happens in the lymph nodes. So that extramedullary hematopoiesis is happening in the liver and spleen early in development, and then also in the lymph nodes as adults. So when we're thinking about this process of hematopoiesis, we're thinking about it from a few different angles. We just talked about where it's happening. We're also thinking about that interplay between different systems, which has to do with the cells themselves as well as the hormones that signal it. So a big idea throughout physiology is the hormones, those drive function. So when we're thinking about the cells themselves, we have to think about different types of stem cells. Remember that generally stem cells are cells that haven't really been differentiated fully and are still capable of undergoing mitosis. When you are an embryo, you have what are called totipotent stem cells. So they have the potential to become the total number of cells that we need in our body. So they can become any type of cell. They have total potential. So a little bit more specific than that are pluripotent stem cells. These have plural potential. So they have lots of options, but they're not quite everything. They are a little bit more differentiated than totipotent stem cells. They're precursors to an entire tissue layer, but they can't necessarily make the whole organism. They can make many different types of cells and tissue. And then multipotent, oligopotent, and unipotent are a little bit more specific. Multipotent refers to multiple potential. It's not as much as plural, but it's a lot of specialized cells within an entire lineage. That's what we're focusing on with hematopoiesis. Oligo means just a few. So like an oligarchy is when a few people are in charge. An oligonucleotide is a few nucleotides put together. Oligopotent stem cells can become a few different cell types. And then unipotent stem cells are fully specialized. They generate more of the same type of cell. So for example, when we talk about oogenesis and spermatogenesis, those stem cells that go through mitosis and replicate themselves prior to meiosis are unipotent. They're making more of that exact same type of cell. So focusing on multipotent stem cells, these are the ones that are again key to hematopoiesis. So when we're looking at a lineage of different types of blood cells, here we have differentiation into erythrocytes, those red blood cells, into leukocytes, the white blood cells, which include lots of different granulocytes and agranulocytes, and then also megakaryocytes, which give rise to platelets, which remember those thrombocytes, those platelets are not actually cells, they're just cell fragments. All of these arise from these multipotent hematopoietic stem cells. And so again, that's just a fancy way of saying that it is having to do with blood, and it can become any type of blood cell. So it's a little bit more specific than totipotent or pluripotent. So we're going to think about these different processes of making red blood cells, making white blood cells, and making platelets. So because we're going to be walking through each of these processes individually, I really don't want you guys to focus on memorizing this whole flow chart by any means. I just want you to understand kind of the relatedness between different cell lineages and also how separate some of them are. So for example, basophils and mast cells are very functionally similar, but they come from different stem cell lineages. So we'll think about the formation of each blood cell type as we go through their function. We're not going to talk about each of these processes all at once. We'll kind of walk our way through each of them and integrate it with function. So before we get into that, I did want to briefly mention some of these growth factors, these signaling molecules that mediate these processes. And instead of memorizing the terms, I really encourage you to again break them apart. This first one, EPO or erythropoietin, you should break it up into erythropoietin. So that poietin is going to be the process of making blood. The IN tells you that it's a protein, a signaling molecule, and then erythro tells you that it's a red blood cell, something to do with red blood cells. So this signaling molecule, this erythropoietin, is secreted by the kidneys. Understanding that the kidneys have a crucial role in monitoring oxygen in the blood is going to be so important for understanding the respiratory physiology as well as homeostasis at the end of the semester. So our kidneys basically act as a monitor for what's in our blood, and a big part of that is monitoring oxygen. When our oxygen levels are too low, special cells in our kidneys secrete this erythropoietin, and that increases production of red blood cells. So kidneys have an endocrine role. They're involved in this signaling and this feedback loop. Thrombopoietin, that thrombo part, tells you that it has something to do with thrombocytes or platelets. Poietin tells you that it has something to do with signaling, the formation of them. So this is a glycoprotein. It's produced by the liver and the kidneys, and it increases production of platelets. And then cytokines, we've talked about, well, if you've already had the immune lecture, then we've already talked about them. If you have not had the immune lecture, we're going to go in depth in them. But these are a huge range of signaling molecules. And specifically a big family of proteins. Some of them are secreted by red bone marrow, leukocytes, fibroblasts, and endothelial cells, so all sorts of cells involved with lymph and circulation and blood, and they are involved in lots of different types of signaling and immune responses. So before we get much further, I just wanted to briefly talk about bone marrow transplants while we're on the subject of creating these cells and the role that bone marrow plays in it. We get to this at the end of the lymph lecture, I believe, some point in the lymph lecture, but I know that that was a lot of material, so you might not have gotten a chance to see it. So when we're thinking about bone marrow transplants, the goal here is to replace some functionality that you've lost in that hematopoietic role of bone marrow. So it's super easy to join the donor registry. They basically send you a swab and they look at something having to do with the surface proteins of your cells. We'll get back to that in just a moment. It's very important to join, and there's a lot of not necessarily stigma, but misinformation about bone marrow donation. So it used to be extremely painful. They would basically insert a very large needle into your iliac crest or near your iliac crest and withdraw that bone marrow tissue. But now it's very simple. Basically, you just collect some blood, isolate those stem cells, and grow them in cell culture. So no need to stick a huge needle into your bone anymore. Just to visualize that process, it's really just like donating blood. They collect that blood and then they're able to isolate and culture the stem cells, transfuse those stem cells into the patient, and hopefully that helps with this bone marrow replacement. to get some of those hematopoietic properties back. But the tricky thing whenever you're considering transplants is that your immune systems have to match. And that sounds very complicated, but really it's a matter of recognizing that the immune system is all about self, non-self recognition, especially the adaptive immune response. We're thinking about what is our cells, what is not our cells, and how do we appropriately attack the things that are not our cells. So that can be a problem if you're trying to get a blood transfusion and you're getting it from someone who has a different blood type. It's the same basic premise. There's different surface proteins, and that leads to different recognition by the immune system and maybe the immune system rejecting that donation. So we have to have that match. And depending on your racial background, your ethnic group, there might be a higher or lower chance of you receiving a donor match. So this this picture is from a story of someone who worked for this news organization. She was a journalist, and she had a problem with her bone marrow. I want to say she had lymphoma. She had something wrong with the rates of mitosis in her bone marrow. And so she was having a lot of trouble getting a donor match because she's South Asian. So historically, it's been notoriously difficult for Black folks and for South Asian folks to get donations. And if you are of mixed race, then it's even harder. So it's important to, one, check like if there's any family members, and two, if you're from one of those underrepresented groups, it's important to become a donor. So that donor matching, you're basically looking at what proteins are found on the outside of our white blood cells, of our leukocytes. And one of those that we test is major histocompatibility antigens, or the MHC complex. We talk about this briefly in the lymph lecture in terms of cell-mediated adaptive immunity, but basically this is a protein on the outside of different types of cells, and it helps present antigens to different T cells. There's some of these MHCs that are found on all cells that have a nucleus. Other are unique to certain types of leukocytes, but they basically, in this donor matching process, look at those surface proteins, determine whether they are going to be accepted by the host or the new host's immune system, or whether they're going to be rejected and attacked by it. So histocompatibility testing is that tissue matching process. And so you're more likely to have particular surface proteins if you are from the same ethnic group as someone. And depending on how many people from your ethnic group donate to these databases, to these donor registries, it really dramatically affects your likelihood of obtaining a match. So if you are of Caucasian descent in the United States, you have about a 97% chance of having a bone marrow match. If you're Latinx, it drops down to 80%. If you're indigenous, it's only 77%. Asian broadly is 72%, but I believe I've read that South Asian is, so like India, Pakistan, Bangladesh is even lower than that. And then people of African descent have a much, much lower likelihood of finding a match. And so this is in the United States. Globally, these numbers are even more challenging to find a match. So this idea of those antigens on the surface of cells, so those proteins on the surface of cells, is the same exact idea as blood typing, whether you're type A, type B, type AB, or type O. Blood antigens display what we call codominance. That means if you have at least one allele for A, that can be like AA or AO, then you're going to have the A antigen. If you have at least one B allele, that means that you're going to have the B antigen. If you have a copy of A and a copy of B, you'll have both A and B. And if you have neither, if you only have the recessive allele, then you're not going to have any of those surface antigens. So basically, your genes are changing the proteins that are expressed on the surface of the cell. Remember that red blood cells don't have nuclei, so those cells themselves are not coding for those proteins, but it's coded for by the that process of hematopoiesis, of erythropoiesis. So those precursor cells to the red blood cells are what's coding for those proteins. So just like with the immune system thinking about adaptive immune response, there are antibodies circulating in our plasma for different blood antigens. So if you are type A and you get exposed to type B blood, then you start producing anti-B antibodies. If you're type B and you get exposed to type A blood, you produce anti-A antibodies. If you're type AB, you don't produce any of those antibodies. And if you're type O, you have both anti-A and anti-B antibodies. That means that those proteins, those antibodies that are floating around in your body, if they come into contact with whatever antigen they're fighting against or targeting, there's going to be an immune response. So if you have those anti-B antibodies and you get exposed to type B blood, there's going to be a very severe immune response. So that's important for recognizing our self-antigens and distinguishing it from non-self. It also means that if you are type A, you can receive type A blood and type O blood, but not anything with that B surface antigen. If you're type B, you can only receive type B and type O blood. Type AB can receive just about anything, and especially if you're what we call AB positive, you can really receive any type of blood. Type O, specifically O negative, is the universal donor. They can give their blood to anyone else and not cause an immune response, but they cannot receive blood from anyone other than O negative. When we're talking about that positive and negative, what we're talking about is something called rhesus factor. So that's another surface antigen, another protein on the surface of the red blood cells, and it's coded for by a different gene, and it's simple dominance. It's not codominance. So if you're Rh positive or Rh negative, those are two different phenotypes, two different outcomes. Rh positive means that you have that rhesus factor D antigen on the surface of your red blood cells, and Rh negative means that you don't. So we usually just shorten this to positive and negative. That means someone who is type A positive, has the A antigen, has the rhesus factor antigen, but does not have the B antigen. Someone who is B negative has the B antigen and no rhesus factor antigen. So when people are pregnant and have a different blood type than their developing fetus, if specifically you have an Rh negative mother or carrying parent and then an Rh positive infant, you really have to be cautious about the maternal immune response, especially if they're planning to have more than one child. So if they get exposed to that first Rh positive infant, their blood cells, because maternal and fetal blood kind of interacts with each other in different ways, if they get exposed to those red blood cells, if they leak into the maternal blood and they get exposed to those Rh plus antigens, then they're going to start producing what we call anti-Rh antibodies. So those are antibodies that are attacking the Rh antigen. They're basically saying, hey, this is something that doesn't belong to our body. So these Rh antibodies are going to be produced and destroy fetal red blood cells, and it's going to be especially dramatic if they have future children that are also Rh positive. So remember, there's that primary response where the immune system is learning. Then there's a much faster and much more extreme secondary response where the immune system is already learned, already produced those antibodies and has them ready to go. So if there's no treatment, then the mother's antibodies are going to destroy those fetal red blood cells. But if you go through this process of anti-Rh antibody treatment, you're injected with a particular protein, this immunoglobulin. That means that you basically have the synthetic response already and destroy that fetal antigen in circulation before there can be any immune response. So you don't attack, you don't have the sustained attack that's you're having to worry about against the fetus. You get rid of that antigen and you inactivate it before there can be any immune response on the part of the carrying parent. So just to help visualize that, if you have your first pregnancy with the Rh positive fetus and there's no treatment, that means at the second pregnancy, there's going to be those antibodies in circulation. And so those are going to attack and destroy fetal red blood cells. But if you do have that treatment, if you anticipate this, and this is again specifically with an Rh negative carrying parent and an Rh positive fetus, if you can anticipate that and you can inject her with the immunoglobulin and make sure that those fetal antigens are inactivated, then she's not going to build up her own immunoglobulins, and so it won't be a severe response with the next pregnancy. So when thinking about the structure and function of erythrocytes, they have a lot of interesting things going on. Like I mentioned, they don't have a nucleus, they also don't have mitochondria, and they don't have an endoplasmic reticulum. Their cytoplasm is basically packed with hemoglobin, and they also kind of have a reduced lifespan and reduced capabilities as a result. They're just super specialized for what they do. Because they don't have mitochondria, they have to go through anaerobic respiration. They can't use aerobic respiration, which requires mitochondria in eukaryotic cells. They also don't have an endoplasmic reticulum, so specifically with the rough endoplasmic reticulum missing, they can't synthesize proteins. They are, however, biconcave. They don't have these bigger organelles inside of them, so they're a little bit flattened in the center. And so that means that they can really maximize surface area to volume ratio and squeeze through capillaries quite easily. That surface structure facilitates gas exchange, and it also allows them to kind of change. They have a unique cytoskeleton, and they can really get through a lot of different spaces. So if you have a situation like sickle cell anemia where you're changing the structure of that cell, it can lead to a lot of problems in their function. When we're thinking about how red blood cells are produced and kind of that hematopoietic process, again, it starts with a stem cell. It becomes a little bit more specialized. It becomes what we call a proerythroblast, so a pre or intro to being a red blood cell, and then you go along the direct developmental pathway. At one point, the nucleus is going to be ejected. Remember that it starts out having a nucleus in these earlier stages of development, but then the developed red blood cell does not have one. The stage right before fully specialized erythrocyte is called a reticulocyte. That is the precursor, the direct precursor to a red blood cell. So this is again happening in red bone marrow. Red blood cells have a life of about 120 days before they are removed from circulation and cleared from our body. And more than 2 million cells are produced per second. So it's not like we produce our cells on day 1 and then produce a whole new set on day 120. There's a constant supply of these cells being produced and taken out of circulation. And so another measure that we can take in addition to the hematocrit is the reticulocyte count. So that's kind of what proportion of our red blood cells are these precursor cells that are ready to form new ones. So on average, it's about 0.5 to 1.5%. If it's too low, then that means that you're not reproducing your red blood cells quickly enough. So it speaks to the potential power of us to have new, fresh red blood cells. When we're thinking about the structure of hemoglobin, so hemoglobin is a 4-part protein. It has those 4 globin proteins with heme groups in the center. So there's these 4 chains of the globin protein. Heme is a special red pigment. That's what gives blood its red color, and it has a central iron ion in it. That iron ion, the Fe, is key for carrying oxygen. And because each heme group on the hemoglobin can carry an oxygen molecule, there's 4 oxygen molecules being carried per hemoglobin. Each red blood cell contains about 300 million hemoglobin molecules, hemoglobin proteins, and that means that you can carry up to 1.2 billion oxygen molecules. So it's just like a crazy amount of oxygen, and you're really maximizing the carrying capacity of the hemoglobin as well as the red blood cells. When hemoglobin is broken down and digested, when the red blood cell comes to the end of its life, the globulins or the globins get recycled in and broken down into amino acids for protein synthesis. The iron is reclaimed, and then the remaining goes towards bile. So this diagram is just showing the complete life cycle of the red blood cell. That's kind of extra information, and it summarizes a lot of information on the previous slides. You should know that certain vitamins and minerals are very important for erythrocyte formation. Iron, obviously, there's a central iron group, but also copper and zinc and B vitamins are all crucial for the formation of erythrocytes. And like I mentioned on the previous slide, those globin proteins, the heme pigment, and the central iron all follow different breakdown pathways. I briefly mentioned earlier sickle cell anemia. This is a genetic condition which happens because having the heterozygote state actually gives you a competitive advantage against contracting malaria. That means that there's this trade-off. It's worth risking having 2 copies of the recessive allele and having sickle cell anemia if you have malarial protection. So the sickle cell anemia, if you have 2 copies of the recessive allele, it changes the structure of your red blood cells. And because you have these cells that are shaped like sickles right here and over here, those get trapped basically in your smaller blood vessels and prevent microcirculation. That microcirculation, actually getting into capillaries and exchanging with tissue, is what really allows you to get oxygen to your cells and remove carbon dioxide. And if you don't have that, then that's really going to interfere with function. It's going to lead to anemia, which, remember, is represented by a very low hematocrit value, and also by kind of this decline in oxygen and buildup of carbon dioxide. In the hematocrit, we also talked about polycythemia. That's when you have an elevated red blood cell count, usually due to dehydration, but also to high altitudes and bone marrow disease. So shifting gears away from erythrocytes and getting into leukocytes, which we see kind of over here. So all those guys are different types of leukocytes. Leukocyte is a very broad term referring to white blood cells. So that's basically all of these other cell types other than red blood cells and platelets. So these tend to be much larger than erythrocytes on average. Some of them can be a little bit smaller, but usually they're pretty large. And they're what we call complete cells. They have all of the organelles, so they're not missing things like a nucleus or mitochondria or an endoplasmic reticulum like the erythrocytes. Platelets are even less complete. They are just cell fragments, basically plasma membranes surrounding cytoplasm. So these white blood cells do a lot of different things depending on what type of white blood cell they are. They're capable of actually leaving the bloodstream and integrating into body tissue to go to sites of infection. So when we're looking at the overall composition of blood, we can look at different metrics. We already talked about hematocrit, so that proportion of blood that's from erythrocytes. We also briefly mentioned the reticulocyte count, so that's anywhere from 0.5 to 1.5% of those special precursor cells to erythrocytes. And then when we're looking at the overall proportion of blood or the overall density of cells within a unit volume of blood, we can see that there's a lot less white blood cells. By cell count, there's a much higher magnitude of red blood cells and platelets, but it's also important to consider that those red blood cells and platelets are much smaller than white blood cells, so it kind of makes sense that there would be a lot more of them relative to the white blood cells. So in normal blood, we see these very distinct types of white blood cells. In cases of leukemia, where you have kind of out of control mitosis, unregulated cell division of those leukocytes, that can lead to very serious complications. And in those cases, we have a much higher value. So again, just like with polycythemia, if you have too much blood cells, it can impair function. So depending on the type of leukocyte, there's very complex and very different life cycles. All leukocytes begin their division process and maturation process in bone marrow, but secondary production and maturation can occur in different locations, such as the thymus and different types of lymphatic tissue. Most leukocytes can live maybe hours or days, but some of them actually end up living months or years. And some of them are also capable of mitosis. So for example, some B and T lymphocytes might be capable of mitosis. So broadly, when we're talking about leukocytes, we divide them up into granulocytes and agranulocytes. Cyte meaning cell, granulo referring to its granular appearance under the microscope. Basically, if it looks pretty spotty, it's a granulocyte. If it looks a lot more smooth, less spotty, very clear, distinct nuclei, it's an agranulocyte. So these end up being very important for both innate and adaptive immunity. We'll work our way through each of these individually. Lymphocytes usually get grouped in with agranulocytes because B and T lymphocytes are agranulocytes, but natural killer cells or NK cells are actually granular lymphocytes. So this kind of structural microscopic classification doesn't really provide a full picture in terms of the different cell lineages, but it is kind of broadly how we distinguish between them. So in the blood lab, you'll get a lot more practice identifying these under the microscope, but you should roughly be able to identify them, especially for lab exams. So we're going to walk our way through the granulocytes. Neutrophils have kind of small granules, but there's lots of them. They're tiny, so there's a lot of them. And these consist of about 50 to 70% of, or these comprise 50 to 70% of the white blood cells in your body. So most of your white blood cells, most of your leukocytes are going to be these neutrophils. And they have this really unique multi-lobed nucleus. So not just two lobes, but many lobes, where their nucleus basically has these round portions, these lobes. It gets narrower and then it goes out again. It gets narrow and then it goes out again and again. So that's what we mean by the multi-lobed nucleus, is all those different structures, and then there's lots of small granules. So these are phagocytotic. They can undergo phagocytosis. They chase and engulf different cells. They're very efficient. They can produce a lysozyme to help break down those pathogen walls. They produce proteins called defensins to help puncture those pathogen membranes and rip them apart. We change our neutrophil levels quite often. They are increased during periods of stress. So if you have a long-term stress response, you have higher neutrophil levels. If someone sustains burn injuries, they usually have elevated neutrophil levels. And during pregnancy, your neutrophil levels also rise. Not necessarily just correlated with stress, but also as a mechanism associated with pregnancy. So this idea that depending on our physiological state, our differential white blood cell count can change is important to interpreting patient data and understanding why they might have different levels of white blood cells. Eosinophils are another one. They have a very pinkish appearance when they're stained and put under a microscope, and they only have a couple of lobes on their nucleus, but they have very dense pink granules. So they have that pink appearance. You can't see through them to the back of the slide as easily. These are really important for producing antihistamines and also breaking down parasitic worms. So a higher eosinophil level, higher than baseline, is usually associated with an infection of parasitic worms. They can act phagocytotically, but they usually don't, so we usually don't group them in with phagocytotic cells. Basophils make up a smaller portion even than eosinophils. They're only about 1% of white blood cells, but they are functionally very similar to mast cells, which we'll talk about in the immune system or lymph lecture. They are slightly smaller than neutrophils and eosinophils. They have much larger granules that are kind of more spread out, and they have a two-lobed nucleus, which isn't as distinctly lobed as the eosinophil. So here you see how the eosinophil really narrows between the two different lobes. With the basophil, there's one lobe right here and one lobe right here, but it doesn't narrow as much right here as the eosinophil does through there. So there's two lobes, but it's not as dramatically lobed as the eosinophil. That's how you recognize it. It's also a lot more purple. So the granules are much larger than the neutrophil. It's a different color and different lobe shape than the eosinophil. It's important for intensifying the inflammatory response. It releases histamine, also heparin. So the histamine helps with inflammation. The heparin stops blood clotting. So it's an anticoagulant. It helps kind of improve blood flow. So this is similar to the mechanism of action for mast cells. Basophils and mast cells, though, come from different hematopoietic lineages. So then we have lymphocytes. These are like B cells and T cells, so B plasma cells, B memory cells. There's lots of different types of T cells, and then there's also those natural killer cells, which are granular, remember. So these lymphocytes tend to be a lot smoother. They have a very distinct smooth nucleus, but they don't have those granules that we see in granulocytes. So we cover these in a lot more depth in the immune physiology lecture. They comprise about 20 to 30% of white blood cells, so a big fraction, but there's many different types of them. There's two types of B cells and many different types of T cells. There's also these monocytes. Remember, if you think back to the hematopoietic lineages, these were a different lineage. These are about 2 to 8% of the white blood cells, so they are precursors to macrophages. So they are really important for having this ready access of phagocytotic cells. They're responsible for engulfing and breaking down debris, pathogens, old erythrocytes, and other cells at the end of the life cycle. So when we're thinking about all the different types of leukocytes, all the different types of white blood cells, we can actually take a measurement of those and then compare our values to a normal range of values. And remember, those normal ranges change depending on whether you have pregnancy, have cancer, have different health situations, then you would expect to have different white blood cell counts. But once you kind of know your baseline and know your expected normal range, you can look and see if your white blood cells are elevated, which are associated with different types of infections or conditions. So for example, neutrophils, that big fraction of white blood cells, elevated levels are often associated with bacterial infections or stress. Elevated levels of lymphocytes are associated with mononucleosis or mono, pertussis, whooping cough, and viral infections. They're also associated with lymphoma, so different cancers. Monocyte elevated levels are associated with malaria, tuberculosis, and fungal infections, so some of those kind of longer-term serious chronic conditions. High eosinophil levels, like I mentioned, are associated with parasitic worms, also with allergic reactions and autoimmune diseases. Remember that eosinophils secrete antihistamines, and so that's associated with some of these kind of broader immune responses. And then basophils, which should be just a very small fraction of your white blood cell count, elevated levels indicate different cancers, chickenpox, and hypothyroidism. So when we're thinking about white blood cells getting where they need to go, if we have damage to tissues, And we have pathogens. Different cell reactions lead to chemical signals being released into the blood. There are a lot of these signals right next to the site of injury, and then they establish basically a concentration gradient. So they are basically establishing a trail, a chemical gradient that our white blood cells can follow to get to that site of tissue damage or infection. So white blood cells do something called emigration or diapedesis. It's basically how they travel through the vessel, integrate themselves into the endothelial tissue. They kind of like squeeze through the capillary walls and then get to where they need to go using something called positive chemotaxis. So this positive means going towards, wish I had a pen that worked, and then chemo is chemical, and taxis is basically like a taxi, it's movement. So these are going towards the chemical instead of away from the chemical. They're following that chemical trail. Some white blood cells hang out in place, they remain fixed. Others are wandering around already in the cardiovascular and lymphatic systems. They're named a little bit differently depending on their function, but these white blood cells that can move around can respond to these signals. So once they're there, they can start doing interesting things like releasing cytotoxic chemicals or going through phagocytosis. So to kind of walk you through this in a little bit more detail, the next few slides talk about this process of emigration or diapedesis. Again, this is referring to general tissue damage and opening up portals of entry for pathogens. We're assuming in this case that our blood vessels are not necessarily damaged. So the first step are those leukocytes migrating to where they need to go. They go through that positive chemotaxis, and then they end up kind of sticking to these special proteins on the endothelial cells lining the blood vessels. So they kind of get to where they need to go, and then they stick in place. They kind of anchor onto the inside lining of the blood vessels. And then that positive chemotaxis continues. They make their way through those endothelial cells. Remember that different capillaries are different levels of leaky. So when we're kind of looking at those openings between the cells, the endothelial cells lining the capillaries or the vessels, we can imagine those white blood cells kind of squeezing their way out and following that chemical trail. So they squeeze through those endothelial cells. They are kind of still like using different protein associations to anchor in place and get to where they need to go. And so they're still following that chemical trail. So then once they get there, they can differentiate into different cell types. So for example, those monocytes can differentiate into macrophages. So we have more phagocytotic ability. Eosinophils can release cytotoxic chemicals. So they can either secrete these different chemicals or act to directly break down the pathogens. So this speaks to this more defensive capability that white blood cells have, where they're acting to remove some foreign invader. This tends to be a more non-specific response. So this is kind of like there's definitely something there, but we don't have an immune history with it. We don't have necessarily antibodies and antigens. It's more like there is a foreign invader. We're going to take care of the damage. These are cells that we were born with. It's innate. And so it's just kind of a non-specific response to get rid of the damage. So when we are thinking about the blood vessels themselves getting damaged, not just the surrounding tissue, but the vessels, we really have to take into consideration thrombocytes, which is a fancy word for platelet. And even though it has that term site in it, they're not actually cells. There's basically a precursor cell called a megakaryocyte, which is derived from a particular stem cell lineage. Remember that we talked about the chemical signaler, that growth factor thrombopoietin, which is secreted by the kidneys and the liver, that acts on those myeloid stem cells, which then differentiate further, eventually becoming megakaryocytes and then breaking apart into platelets. So they're basically fragments of the cytoplasm in the membrane that are involved in clotting. They are basically stored up in the spleen, and they are really important for limiting the amount of blood that might be lost. In the hemodynamics lecture, we talked about blood homeostasis and hemostatic mechanisms of slowing blood loss. So these thrombocytes are really important for that process. If you have too many of them and you have too much clotting, then you have thrombocytosis. If you don't have enough of them, then you have thrombocytopenia, and that can lead to abnormal bleeding and hemorrhaging. Again, remember that thrombopoietin is a hematopoietic growth factor that we talked about earlier in the lecture. As always, whenever there's hormones, I encourage you to make flashcards just so you can associate the term with the function. So I mentioned that these thrombocytes are crucial for this process of hemostasis. So that term does sound very similar to homeostasis. Homeostasis is more broad. Hemo should make you think about blood. Stasis is being consistent, standing still. So hemostasis is the process of making sure that our blood stays where it needs to be. This is going to be really important for a written question, so make sure you know the 3 main steps of this and that you're able to summarize them in a sentence or less. So generally, when we're talking about hemostasis, we're talking about this process by which we have blood clots forming. And in this image on the right, that is what we see. So basically, this image over here is showing erythrocytes, those red blood cells, and it's also showing this special mesh, which is catching different material and helping to prevent blood from leaving the vessel. The implication there is that there's some damage to the vessel, some vascular damage that is causing blood loss. So if we're not able to go through this process of hemostasis, that leads to hemorrhaging. So if we have an injury to a blood vessel, again, not an injury necessarily to just body tissue or epithelial cells, if the vessel itself is severed, then we start this process. And the 3 main steps are the vascular spasm. Something is happening to the blood vessel, the muscle is contracting, and that's stopping blood from leaving the open vessel. Then we have platelet plug formation, where basically it's the first kind of activation of those platelets. They start to stick together and form a platelet plug. And then you have proper coagulation. There's a change in the proteins that leads to that special mesh, and it really forms a proper clot. There's a lot of different kind of pathways that can be involved in this. The most important part that is similar through all of them in terms of the cell biology is a conversion of proteins, specifically fibrinogen to fibrin. We talked about fibrinogen as a plasma protein earlier in the lecture. So we'll get back to these different pathways in just a moment. So again, walking through those individual steps, the first one is hemostasis. Step 1 is vascular spasm. That's what you see in this image. So we see the muscle contracting. This is the spasm where you have that narrowing of the blood vessel, of the vasculature. So this is done because when the blood vessel is injured, these special proteins, these signaling molecules called endothelins are released, and that causes the smooth muscle to contract. We kind of talked about this idea in terms of autoregulation when we were talking about hemodynamics. So this endothelium can release different signaling molecules to autoregulate, to cause that blood vessel to contract so that you're not losing any more blood. So then we have the platelet plug forming. There's a special plasma protein that we haven't mentioned yet called von Willebrand factor that helps with this platelet clumping. So there's a whole cascade of reactions that happen. The platelets clump together. They also release ATP, so that's responsible for helping to reinforce the platelet plug with more platelets. They also releases serotonin, which maintains that vasoconstriction, that vascular spasm, and then prostaglandins and phospholipids, which are also responsible for vasoconstriction and activate more clotting chemicals. So this platelet plug is like a temporary band-aid. You're getting the actual platelets clumping together, you're physically blocking the hemorrhaging from occurring, and you're also releasing a bunch of chemicals that help reinforce that and keep that muscle spasm going. So then finally you have the coagulation cascade, and this is where you get a proper clot. I'm really sorry, my dog is snoring so loud right now, so I'm sorry if you can hear her. So like I mentioned, fibrinogen is that precursor blood protein. It's that plasma protein that we have in circulation. When we have a clot and when we have a platelet plug and we have that series of chemical signals, that is converted into fibrin. And that fibrin, it's a fiber protein. It forms basically a big mesh that traps platelets and blood cells and all kinds of material to form a stronger clot. So it's not just a platelet plug, it is a proper clot. And this process is mediated by many different clotting factors. So some of those include fibrinogen, prothrombin, calcium ions, and vitamin K. Vitamin K is especially important for clotting. Your textbook has a whole list of clotting factors, and in nursing school you might need to go into detail on knowing all of them. You're not going to need to know them for this class. You should just generally know that relationship between fibrinogen and fibrin, these different steps of hemostasis, and kind of broadly why it's important. So there's different pathways, different ways that we get to this coagulation cascade. On that image earlier, I showed you the extrinsic pathway. That is due to trauma. The intrinsic pathway is due to internal vessel damage. So you can have external trauma that initiates the extrinsic pathway, or internal damage, which initiates the intrinsic pathway. Regardless, both of those end up with this common pathway in which that fibrin is produced, you get the mesh, and you have the actual clotting and coagulation occurring. So the tricky thing though is that just like how you can have too many platelets, you can have too much clotting. And if you have those clots forming, they can be released into your bloodstream, end up somewhere else, and block the flow of blood where it needs to go, for example, to your brain. So that's very serious. So we want to avoid having too many clots forming, and when we do have clots, we want to eventually break them down when they're no longer necessary. So there's a couple of different mechanisms. One of them is the gradual degradation of an existing blood clot. The other is the prevention of unnecessary clots. So fibrinolysis is what we are talking about when we have an existing blood clot and we are lysing it, or splitting it apart. And it basically has the opposite series of reactions as the hemostasis. So there's vasodilation instead of vasoconstriction. You're stopping that spasm, you're opening up the blood vessel again so the clot can be released. Plasminogen is a different protein that's converted to plasmin, and plasmin acts to break down fibrin. So it basically stops the spasm and creates a protein that breaks apart that mesh, that frees up those different cell or clot components, which can then re-enter circulation or be broken down further. Plasma anticoagulants also circle around. That includes heparin, like we mentioned earlier, from basophils and mast cells, a anticoagulant called antithrombin, and other substances that just generally circulate and prevent this reaction from happening unless you really need it and you have vascular damage. So these help to make sure that the clot only forms when you do truly have vascular damage, when you have vascular injury. Okay, so I wanted to kind of shift gears a little bit and think back to that idea of kidneys kind of monitoring blood, and also how blood might carry respiratory gases. So again, this is kind of wrapping up blood physiology and setting us up for respiratory physiology. So this is a slide that we saw earlier about the structure of hemoglobin, just kind of reminding you that hemoglobin has a tremendous oxygen carrying capacity. So again, a key function of erythrocytes of red blood cells is that they are capable of carrying respiratory gases. Traditionally, we think about that as being oxygen, but it's not just oxygen. So when we think about this binding occurring, remember that there are those 4 globin proteins. Each of those has its own central heme group. And so as we're looking at this GIF, we see that oxygen has a little bit of trouble binding to the first globin unit. It kind of binds to it, and then there's some change in the shape, and that means that each subsequent oxygen binds even more quickly. So that first oxygen is binding to the subunit. The second oxygen is, then there's a conformational change. Second oxygen binds more quickly, another conformational change. Third, even more quickly, conformational change. And then the fourth oxygen binds and hemoglobin is fully saturated. So that measurement of how many of these hemoglobin are saturated is going to be very important clinically and in terms of understanding respiratory physiology. I'm introducing it to you now because we are going to go over some pretty tricky things with oxygen saturation and the curves and like kind of the relationship between how readily hemoglobin lets go of oxygens and other factors. So I want to introduce it to you now. So when we're thinking about oxygen saturation, what we mean is the percent of total hemoglobin that are fully saturated with oxygen. So we're comparing these guys over here, which are hemoglobin but are not saturated or unsaturated. So they do not have oxygens on their heme group versus these guys that are saturated. And so we understand that there's that relationship between kind of the flow of oxygen, having those blood cells in the pulmonary circuit pick up that oxygen and take it away and remove it from the lungs is important for maintaining a concentration gradient and making sure that oxygen is flowing from the alveoli into circulation. If the oxygen didn't bind readily to the hemoglobin, then we would not have that concentration gradient be so strong and it wouldn't be coming in from the lungs. So the actual measurement of oxygen saturation, and we'll get through how to calculate that in just a moment, is about 95 to 99% in a healthy adult. And because we have that clear metric, that's something that we can actually measure. So like I mentioned when describing this GIF, there's a relationship between the percent saturation of hemoglobin, that oxygen saturation measurement, and the partial pressure of oxygen. And there's lots of external and internal factors that affect it. So I just want you to be familiar with that idea so that when we go over respiratory physiology, it makes a little bit more sense. So the way that we actually calculate oxygen saturation is by looking at the fraction of saturated hemoglobin out of all of the hemoglobin. So that oxygen saturation measurement, also called SO2, which is measured as a percentage, is the saturated hemoglobin divided by all of the hemoglobin, unsaturated and saturated, and multiplying it by 100 to get a percentage. So again, the unsaturated hemoglobin are the ones that don't have oxygen at all 4 globin proteins. The saturated ones do. So when we're looking at oxygenated or saturated hemoglobin versus deoxygenated, the color of blood changes a little bit depending on the chemical properties of hemoglobin, whether it's saturated in oxygenated blood or relatively unsaturated in deoxygenated blood. When we say deoxygenated, we don't mean that there's no oxygen whatsoever. We just mean that there's a lower partial pressure of oxygen and less hemoglobin that are saturated. So the saturated hemoglobin we call oxyhemoglobin. The unsaturated we call deoxyhemoglobin, or if they're bound to carbon dioxide, carbaminohemoglobin. So when we measure this in a lab, we know that the color of those blood types is a little bit different. If there's a lot of the oxyhemoglobin, it tends to be that bright red crimson color. If it's deoxyhemoglobin, it tends to be a darker red. And because there's clear properties of light involved, we can actually measure that using a technique called pulse oximetry. It's non-invasive. You can easily put one of these probes on a patient as long as they're not wearing nail polish. And so it's basically relying on the ability of different chemicals to absorb light differently. So those different chemical properties of oxyhemoglobin and deoxyhemoglobin, and looking at the absorbance and quantitatively measuring oxygen saturation. So we put this simple pulse oximeter, this simple probe on, it shoots out different light, the machine measures and processes it, and we get a value. That's really easy to do if you have someone who is asleep, someone who's in a ward long term. So it's useful for diagnosing sleep disorders. It's useful for intensive care, just kind of like routine screening. If someone has persistent cardiovascular, cardiopulmonary conditions, it's good to use. So in different hospital wards and emergency medicine. And then also pilots and climbers tend to use it because high altitudes affect our oxygen saturation. It's also important for athletes. So what pulse oximetry is doing is helping us measure hypoxemia. Hypoxemia specifically means that you have a low amount of oxygen in your blood, less than 95% oxygen saturation of hemoglobin, or arterial partial pressure of oxygen in a range of 80 to 100 millimeters of mercury. So that arterial value is a little bit harder to actually measure. It's hard to go in and take arterial gases, but the pulse oximetry is a useful measurement. It's very hard for us to measure hypoxia. That means low oxygen in our tissues. Instead, we measure hypoxemia because if you are hypoxic, you may or may not have hypoxemia. If you have hypoxemia, we can easily measure it, and you for sure have hypoxia. So like I mentioned earlier, the kidneys are responsible for sensing and responding to that hypoxemia. They basically sense that change in the oxygen within our blood as they filter the blood. As we have that dip in oxygen, as we have hypoxemia, the kidneys and the liver sense that and secrete erythropoietin that stimulates the red bone marrow to produce more red blood cells, so it accelerates erythropoiesis. You get a higher red blood cell count and an increased ability to transport oxygen. So we'll look at a respiratory physiology, during respiratory physiology, we'll look at an altitude sickness case study and talk about this quite a bit more, but this is a negative feedback loop. It doesn't go on indefinitely. You don't want to keep producing red blood cells because then you get polycythemia. So at a certain point, as your oxygen saturation levels rise again, you drop that amount of erythropoietin that is being secreted, and so it's a negative feedback loop. The effect results in the end of the stimulus. Okay, so as soon as I post this, I'm going to also post an online lecture quiz. It will be available from today on Wednesday, March 4th until Wednesday, March 11th at 11:59 p.m. It's covering cardiovascular physiology, so anatomy, EKG and the cardiac cycle, and hemodynamics. Please make sure you look it over. And if you ever miss an online quiz, you should absolutely still do it. I don't close the quizzes. You need to be able to see the questions so that you're prepared for the exam. So the material is always there for you. It's just up to you to actually go sit down and look through it. Just like I can't force you to come to class, I can't force you to watch a video or take a quiz, but I strongly encourage you to do so for your own education. So this lecture, again, is really important for the lymph lecture and also for respiratory physiology. The online lectures are not any less important than the in-person lectures. Please make sure you're making time for them and let me know if you have any questions at all.